Hello, everyone. Welcome to the. And we're really excited to have all our speakers today and to have all of you joining the session. So before we begin, I'd like to say that there is a chat that you can access on the top right of the website, and you can ask questions there. So we'll take questions from the chat and hear the speakers' answers. And so now I'll hand it to my co-chair, Leonora, to introduce our first speaker.
Everyone, thank you today for this pre-conference session. The next generation session initiated by the OTS to address the younger generation, as well as highlight some of the fantastic research work of younger trainees. And we have a really exciting lineup of talks coming up for this. In addition to that, we're also very excited to highlight our keynote speaker for this session, Professor Keith Gagnon. Keith at Southern Illinois University, where he shared between the biochemistry and molecular biology, as well as the chemistry and biochemistry departments there. And today we're going to get a look into some of the more personal aspects of Keith's life throughout and how he got to be where he is today. He grew up in New England and married his high school sweetheart, and they have. Keith enjoys basketball, art, working on cars, and home improvement projects. Also not included in his bio, Keith enjoys shredding the OTS party dance floor with other OTS members, and I'm sure is looking forward to a time when it'll be safe to go back to in-person meetings for this reason. He did his BSc and PhD at North Carolina State, and he conducted the research for his dissertation with Professor Stuart Maxwell, studying RNA-guided small nucleolar RNA and RNP enzyme structure, function, and evolution. He then went on to do a postdoc in Dallas with Professor David Corey at UT Southwestern, where together they worked on human nuclear RNA interference and therapeutic nucleic acids for disorders. And then in 2014, he started his own lab, where the focus there is on molecular mechanisms and therapeutic strategies for repeat expansion disorders, specifically ALS. His lab also focuses heavily on CRISPR chemical biology and guide RNA modifications for therapeutics there and understanding the structure-function relationships there. And other projects in his lab include basic research on non-coding RNA mechanisms. And this last year, his lab has been highlighted significantly for their efforts in the fight against COVID-19 and identifying new SARS-CoV-2 variants. And Keith is no stranger to the OTS community. He's been attending OTS meetings since 2010, and he got involved in the Scientific Advisory Board and Board of Directors after starting his independent position at SIU. And in addition to all of this, Keith makes every effort to welcome new members and support trainees, which is why he was for the Next Gen keynote session. And we're very proud of him. So Keith, I'll pass this over to you, and we look forward to seeing your presentation.
Well, thank you guys so much for the invitation. It really is very much a pleasure and an honor to be a keynote speaker for this session. Yeah, great introduction. I appreciate that. I can't wait till we can hit the dance floors again. So hopefully by next year, who knows what this pandemic will give us. What I want to talk about today, being that this is a next gen early career session, I want to talk about science, but I also want to talk a little bit about what also happens to us while we're doing our science and pursuing our careers. And hopefully my title gives you, you can pursue lots of things as my lab does. We have a common thread of RNA, all that. But importantly, you should really enjoy what you do. You spend so much time in science, try to make the best of it and make it fun. So good science takes time. It takes a lot of hard work. And that's just the reality of it. But you should also try to enjoy it. And so we have generic motto in the lab, hard. We take our science pretty seriously, business, but we really want to have pizza for lunch once in a while and go bowling and fun. So this is an important theme, hopefully to take away. This is a brief overview of kind of the things I'll touch on, and it's a little bit about how I got here, and my story. You guys will have your own stories, and your stories growing and evolving over time. And so, years down the road, you'll be able to look back at how did I get here? How I got to grad school, what happened to my PhD, some about the postdoc years, and then what's been going on since I started my lab and some different directions we've taken. Okay, so I grew up in a New England working class family. So I was the first kid in my family to go to college. My dad was a carpenter, my mom was a homemaker. So this idea of being a scientist was, I don't know, kind of a dreamy thing when I was a kid. My mom says I wanted to be a scientist. That I don't remember saying that, but what happened. Even had a school photo where I dressed up, I think, once. Ended up going to NC State for I moved to North Carolina in high school and then went to a local state college in North Carolina, NC State University. While I was there, and I really wanted to get into the lab. I had this wonderful undergraduate advisor, Steve Maxwell. Here he is, Steve Maxwell. And he offered to let me work in the lab. And there I was introduced to small nucleolar RNAs or SNO-RNAs. They were really the first sort of RNA-guided enzymes long before RNAi or CRISPR or other RNA-guided systems were really understood. They were kind of enzymes. So it was fascinating. I decided to stay for what was called sort of a 5-year degree, where for an extra year and a half, you did some graduate classes and you got a master's. So that was the plan. Started my first semester and he took me to a local RNA conference hosted by the RNA Society of North Carolina. Just enamored at all the cool things RNA did. The title of the symposium, Tool and Target. So there was Therapeutics, and it was fascinating and I was sold. So hence I started my PhD. Was pretty much as you would expect. I was immersed into many long hours in the lab, doing all the things you can imagine, purifying proteins, running RNA gels, trying to think deeply about problems and understand the literature. I always liked to solve problems. So I was one of those students who long hours, and I would spend all night in the lab sometimes, not because I was trying to meet a deadline. I just wanted to see what the result was going to be. And I didn't have kids at the time, so I could get away with that. One of the key things I took away from my PhD was I was sort of initiated into the competitive side of science in a good way. I think that was instilled in me, and also a passion for just do good science. Didn't want to do sloppy science. And that's exactly PhD. And here's an example of the kind of gels we used to run, these RNA cleavage, RNA structure gels. So, old school RNA biochemistry. During my PhD, we made some important discoveries, small on the assembly, structure, function, evolution of these boxy RNP complexes. We were interested in the different core proteins RNA, so how they assembled the complex, what that structure looked like, protein RNA complex, and then the evolution. There was some distant archaea, and how did those compare to the humans? And so one of the things I want you to notice is the dates of there long after. In fact, when I graduated with my PhD, I only had 3 papers. I had one first author in JMB, and I had a methods paper and a review, and that was it. But I had done a lot of work, and so I continued to write during my postdoc, put out papers subsequently after I left. So the PhD was great, got my passion for science, but there was other things going on. Honest, there's more to life than science. It's great, that's for. But the things that happen in life can be one of two things. They can either maybe be or they can be something that you sort of embrace and just make part of the process. And that's sort of what I've tried to do. Like was mentioned, I married my high school sweetheart. We bought a house, which was well suited to me. It was a good distraction. I grew up in the summers build houses with my dad. So this was, yeah, maybe I should have been in the lab more, but I was having fun painting and, building. And our first child was born, our son. So there were one outside the lab that you have to balance. I moved to David Corey's lab to do my postdoc at UT Southwestern Medical Center. And by going there, I wanted to learn new tech, introduced to nucleic acid chemistry. I knew nothing about nucleic acid. What RNA was, that was my world. Repeat expansion disorders, we were targeting repeat expansions with chemically modified steric ASOs. And then also RNAi in the human cell nucleus. We did a lot of time work characterizing that process. But one of the more important things I learned in David Corey's lab was what I call the business side of science. And I will always be greatly indebted to David for emphasizing these things. He never had to go out of his way, but it was obvious that, hey, you've got to be a great grant writer. You've got to understand how to manage a lab and manage people, and how and when to submit papers. It was a great experience from that standpoint. So I kind of learned, quote unquote, the trade, if you will. And of course, things were going on. I renovated another house. Now, this picture I'm showing you is the final renovated house. It did not look that nice when we bought it. Lots of hours. And I got to give a shout out to John Watts and some other people who came over, I think, and helped us work on this. Some colleagues and friends. Our second child was born, and yes, all my kids are crazy and funny like this. It's not my fault. That's my wife. She's got the good, funny genes, I think. And something I got into that I never would have expected would help me was I decided to volunteer at a local church and be involved in the community and sort of mentoring in this 12-step recovery program. The point is helping people make good life decisions. Why was this so valuable? It took up extra time. I would say this is the single most important thing I learned to prepare me for mentoring students and helping them make good life decisions, not just in science, but outside of the lab too. It was super important, and I really appreciate that. And I, even though I often feel like the grad students are kids, listen, they're adults, they're young adults, and I was there once too. And we need mentors and people to give us good feedback. There's a lot of different career directions you can go, obviously. And when I was, postdoc, this wasn't even 10 years ago, there were plenty of options. And there's even more now for PhDs, people with good training. But pretty early on, I knew I wanted to be a PI. It was just when I had my heart set on that. And we had this saying in David Corey's lab. It was a colloquialism, like just a saying we would reply to almost anything. When someone would say something, we'd say, "Good luck, you're gonna need it." Well, that's kind of how I felt. It's not. A lot of people set out wanting to be a PI, and you can have good publications. Those things don't guarantee success long term. It can be a tough business, succeeding as an science. But there's lots of ways to succeed. Hopefully that will be a take home here. So what happened my first year of applying for jobs, I sent out about 25 one interview. So I said, okay, I'm going to double down my efforts. Second year, send out 50 applications. Got a few interviews, I got one offer. So, it's, by the way, this is kind of the experience you'd be for many people who you think well qualified. They've got a great CV. It's there's only so many faculty positions. So the one offer I got was the current place I'm at right now, University School of Medicine, and obviously I took that job. But I had two decisions to make. Do I, because it's you may not have heard of SIU. It's not a big visible school. That try to build my CV, try to publish another paper, go again next year. That's not promised. Who knows if that paper is going to get out in time? As I accepted this, and I took the leap with some good advice from my PhD advisor. So when you start an independent faculty position, you'll all of a sudden be confronted with many things beyond just, hey, doing cool science. By the way, you need to teach. Oh, yeah, you want to do experiments? Well, you're the only one who knows how because nobody's trained. Even if you hire a new postdoc, there's going. So you do the experiments, you train, you write the grants, write the papers, you submit them, and then you rinse and repeat. And it can become tiring and even wearisome. And but again, if you do this, and I'm going to give you some just sort of tips and tricks and things that I've learned over the process that I think have helped me. So some of the challenges I faced early on is I entered my first year and I need to teach this massive undergrad biochem class, 100 students, 5 lectures. Sure, I'm a biochemist, but I haven't taught this before. So how do well, I contacted my PhD advisor who had taught lots of biochemistry. He sent me some lecture templates, and I'm showing you a key because he always had a sense of humor. He said, Keith, I'm going to send you the keys to the kingdom. And so he sent me the keys. He about saved my life. I would have been just sunken under, creating lecture. So, used my resources. Low startup. Out of an institute that had endless resources. I needed to get grants. And so my first year, I luckily was quite fortunate. A CDMRP grant, defense grant for our ALS work. The first 2 years, so that was great. Next problem, who's going to do this research besides me? Because I left 3 good students right off. They're all now graduated. Zach is at UMass doing a postdoc with John Watts. Kushal is in California. She's working in industry and she's having a great time. I think between you and me, I think she makes even more money than I do, so she's doing fine. Iman she went and got a competitive Saudi starting in MIT for a postdoc fellowship. So happily, they're doing well, and I got a good set of students to start out. Next problem, well, you got to publish. I don't have any data. So one of the solutions is you try to take some data and stories that are unfinished and try to finish them in your new lab. A crystal structure that we had worked on in his lab. So I had led that project. David, generous, he let me be the corresponding author. So again, having good advisors and mentors, people looking out for you was very helpful for me. Last thing, no lab space. What? I just got a faculty position. They're not going to give me a lab? Well, it had to be renovated, and it took about a year to renovate. So I had to squat in other people's labs and borrow lab space. That's okay. Don't let that stuff slow you down. Got a beautiful, nice lab space, and good mentors or great mentors will produce great mentors. I had good mentors. I had a great PhD advisor. David Corey was a great postdoc advisor for me. You don't always agree with them at the time. You don't always follow all their advice, but darn it, they're just appreciated later down the road. One of the things my PhD advisor told me, he would say when I would have doubts about, making it in science, he would say there's always room for good scientists, no matter how bad the climate gets. Some other, and by the way, I'll mention these are some quotes that I recall. These people probably may not even remember saying these things, but it's just amazing how in life, sometimes people say something, even just in passing, and it sticks with you and it changes you, and they might not even have noticed it. One thing my PhD advisor would say, he said, pick one thing and be great at it. Well, I didn't take that advice. I picked a bunch of stuff. I'm trying to be good at a bunch of stuff, but that's because I just have a lot of good ideas, I think, and I want to pursue a lot of things. Another quote that I recall from him, I'm trying to decide if I should take this first job offer at SIU, and he says, Keith, do you want to be a faculty member or not? Yes. So that was easy. David Corey, I remember him saying this, and it just stuck with me. He said, success is the best teacher of success. You want to know what success looks like? Look at people who've been successful. More practical advice, though, he told me, is don't write a lot of reviews when you start your lab. They don't count toward tenure. Thank you, David. Took that advice. I published a few. And other practical things from department chair. I had this funny colleague, and I was complaining about grant writing once. He said, it's not the grant writing I hate, it's the success rate, which is true. Actually, grant writing is a fun process. And one of the best things I've done is team up with good people and collaborate. Many of you know Masad Dama, and he's just got this way about him. And one thing he says, I think he said many times, is let's tango. So we've got these ideas and let's get in the lab, let's get it done. And so I would say surround yourself with people who are encouraging, who are going to stay positive and help guide you in the right direction. Okay, so I'm going to say in the next few slides a little bit of general advice I have about grant writing and publication. I don't want to take up too much time, but when it comes to grant writing, I would say just keep submitting. You got to believe in your ideas even when you get rejected, okay? So don't grow weary of sending in grants. Try to stay excited. Always find time to think. I know it gets busy. There's all these meetings to go to. There's always things to do, but you've got to protect time to think, to write, and dream, okay? Also, I would say don't wait until you have the perfect data to submit. Sometimes that perfect set of data may never come about. You may never get the perfect data. By the time you do, also, it may be too late. The idea might have soured, okay? So I say submit grants early and be aggressive and see if your idea is any good. Last thing, practice makes perfect. Well, you'll never write the perfect grant, probably. I mean, very few of us get perfect scores, but the point is, how do you get better at something? You do it a lot. I've submitted somewhere around 50 grants in the past 7 years. You should be submitting 6 to 8 grants a year at different agencies, exploring different sources, going after funding announcements that seem like they fit you. That's just going to give you the best chance of success, and you're going to get manuscript writing. I don't have a whole lot to say here. Everybody write and just write a good story. But different philosophies. I much prefer writing and publishing as you go and trying to, not every project's going to be a great Nature paper. And you determine that as you go. You could wait 5 or 6, pile up, and ultimately it might not go to that big journal. Sometimes it's better into smaller papers. You have to build that reputation and a success and make the impact you want in a different way. Are your students and the people that work with you and for you when they graduate, so it's to their benefit in a way to make sure they get their names on, make sure you're helping them and giving them opportunity. Last thing is a little bit different, but I would say collaborate, because if you have a story and you notice that you have a gap or a hole, team up with the right people to help you finish out and make a great story. Okay, a few minutes and talk about some different science directions. Pretty diverse. The common theme has been RNA. But for this first project, for example, this has led to 2 good papers. We started out wanting to make cell-based models to do cellular biochemistry. Fluorescent RNA aptamers are a great tool to defend cells, but unfortunately, these broccoli spinach aptamers did perform well for us. But we had invested all this time in developing that, playing with it, and I thought, well, let's understand a little bit more about the structure-function relationship within. One of the features was a magnesium dependence difference between some of these aptamers. I turned to things that I knew were doable, these RNA structure mapping, sequence alignments, fluorescence spectroscopy. And then we added in a new twist with some sort of high throughput mutagenesis. And basically we found that the magnesium dependence came down to purine-purine mismatch. And if you replace that purine-purine mismatch with a Watson at the base of this quadruplex in the structure, you could reduce the magnesium dependence. So, and it was a great little study. It was doable. It fit well for the lab. And we were able to get some publications out of that. Cell-based models, diseases we study to enable, like I said, biochemistry, RNA biology, therapeutic screening and studying. But what we found out pretty quickly, cell-based models are not easy to build. It actually takes time. And one of those things was the repeat expansion disorders we're interested in. We realized we're going to have to learn how to expansions and get and validating them. So we took some time and we developed a protocol. It's an iterative cloning approach. Allows us to drop in repeats, really any desired size in a very predictable manner. And then we turn to nanopore sequencing to sequence and validate these. And what you see down here on the bottom is an example of G4C2 repeated 88 times. It's exactly what we set out to clone. We're able to do it. And so we were really pleased with that. And get closer to building the model. We wait until we build the models and validate them. I don't know, that could take 10 years. So we want to publish as we go in my lab. Okay, so now we're going to talk about CRISPR. Well, when I left David Corey's lab, I wanted to continue working on RNAi. And David kind of warned me. He said, it's going to be tough to get funding there. And he was right. But an interest in RNA-guided enzymes, and CRISPR was one of those. And there was really nothing published in the literature where anybody modified the guide RNA component. And so we set out to do that. Soon after, teamed up with Masad Dama's group, just basically used chemical modifications to probe structure, understand the biochemistry, and ultimately, hopefully, benefit the development of CRISPR. So some good papers out of that, with more recently Cas12a. Okay. Interesting twist, as we're doing all this chemical modification, I started thinking about how some of these guide RNAs can bind the Cas9, but they're not active. And so I thought, well, maybe you could chemically modify some of and make modified oligos that can actually inhibit the CRISPR. Why would you want to inhibit CRISPR? Well, for safety reasons, as a kill switch for therapeutics, etc. So it turns out this seems to work pretty well. We've got an anti-PAM module, which binds the enzyme. Another part acts like an ASO that binds the trace RNA. And together, these give really good inhibition. I won't talk much more about this because my student Chris, in a later session in the meeting, will talk a little bit about our progress towards in vivo, getting these in mice to see how well they work. Project I'd love to talk about, but I can't, was about 2 years ago, I was given the opportunity to help co-found a therapeutics company. I didn't go looking for it, it just came to me. And so it was a nice, things fit well. And the idea was targeting repeat expansions with microRNA mimics to potentially treat these diseases. So far, the data is looking really great. I can't say much more about it. The point is, though, it's a cool opportunity. And sometimes when you have opportunities, the pandemic hit in March of 2020, and we were home. I didn't want to go home and sit on the couch. I wanted to try and do something that might actually, help fight the pandemic. And so we've got this mammalian sequencers, and this thing's not going away anytime soon. As you can see here, it keeps coming. And so we said, well, what if at some point people want to know what are the mutations that this virus is accumulating? And this is before we were calling all that. So we set out to sequence a couple of virus genomes. We've now sequenced well over 4,000. And part of this is partnering with the state of Illinois to do the genomic epidemiology or genomic surveillance. Primarily, we're focused on the spike protein, right, that mediates cellular entry and its mutations, but we can track. And we can also see the distribution through the state and its introduction and how things are moving. So that's been a really, and we've been able to help, so sometimes when opportunities come, you just gotta. Other things were happening during, trying to get tenure. One of the most difficult things was my wife was diagnosed with breast cancer. And, and she became pregnant at the time. So we had our third child was born. My wife went through chemo, and both our daughter and my wife are perfectly healthy now. Great. So for 2 years, that was pretty tough. The beautiful thing about it, sometimes you just gotta, because after all this, I got four NIH grants funded, and so and it all worked out. Just leave thoughts about career. It's not always fun. There are real deadlines. This is a business in some ways, but it's okay if things don't work out. I just try to find. Also, be patient, be persistent, be professional. People are watching. You're building your reputation. And even when you think people are not watching, your colleagues are watching to see how you handle things. Now, the business side of things can really distract you a little bit. And I always try to remember your first love, so to speak, how you got into science, why you're doing science, and hopefully that will help center you. As far as what kind of science to do, take calculated risks and follow my instincts. And my hope is that discoveries will follow. And so that's sort of my philosophy. The last things I want to talk about, not you or me, but about the people that work for you or that you work with, your colleagues, the students. This is not just our career, but we spend so much time at the lab. The lab is like a home away from home. So I like to treat everybody in the lab like we're family. And I want to establish a good lab environment, a good culture that I can 20 years. And that leads to the last point, once you're tenured and things are going good, it's not really about your career anymore. It's a legacy of what are you doing for your trainees and what's sort of their career. Thank, obviously, funding that's made all these are current people who contribute to a lot of these projects. And again, thank you so much for the opportunity to talk, and hopefully I can answer questions or catch up with you guys later in the meeting. Thanks.
Thank you so much, Keith. Here, we often tenures of we don't get to hear the other sort of more personal aspects of life, which are obviously important. We don't have too much time for questions, so I'll just briefly, I wanted to talk, you talked a lot about mentors and how important they are, and I fortunate enough to find really great mentors in your advice. What do you think you could just give like a word of advice to the trainees watching on how they can seek out mentors?
I think good mentors is not just the mentor, but also you. I agree 100% with them. In fact, I was so pissed off at my PhD, but I was ready to graduate. That's how great it was. It was time for me to move on. And David Corey and I didn't always agree on things, but looking back, so part of it is you got to be humble and real everything. So listen to advice, take it into consideration. And also, if you do have the luxury of choosing and picking and choosing, try to pick somebody who you feel like you can get along with and learn from for the next couple of years.
Yep, absolutely. I think that's really important too. Encourage everyone watching Keith in spatial chat after the session. Thank you again, and I'll hand it over to Aurelie for the next talks.
Right, welcome back everyone. I'm going to go to the next part of this session, and we'll welcome all the presenters of the session. So we can go to the next slide, and the next slide. Happy to welcome speakers. The session really aims at trying to give the young generation to showcase their efforts towards expanding in oligotherapeutics. So we will first hear talking about aptamers for COVID detection. We'll then that we'll discuss the synthesis of DNA and RNA oligomechanochemistry. We will then different topic, hearing about uptake of antisense oligonucleotides, different methods. We'll then hear Albert talking about object delivery nano and how we can study that. Finally, colleague Hala, who will discuss the impact of epitranscriptomic modification on so again, feel free to ask questions on the chat. Leonora have 5 minutes from the audience speaker to answer. And if we go to the next slide. Yeah, so yeah, Leonora, you want to?
Yes. So immediately following, give you guys a little bit of a, but then we want to bring you right back to spatial chat where we will kick off our next gen early career social. And what we're going to do is gather there, and then interested in networking can hang around in spatial chat, but then those interested in join in on a little bit of trivia, we'll give you instructions about doing that. So that will kick off at 4 p.m. after Eastern, 9 p.m. if you're in the early, or rest of you, it's a little bit late on that end, but we'll try. So in terms of the social, we'll get right into the talks. We're a little bit behind. So our first speaker is Lucy Yang, and we will give the stage to her.
All right, good morning, everyone. From Seattle. I'm in Seattle. My name's Lucy, and I'm a PhD candidate in bioengineering at the University of Washington. Thank you, Professor Gagnon, for sharing your experience and insights. Today, I'll be presenting on the SARS-CoV-2 spike protein entity binding aptamers for rapid detection. As we are all acutely aware of, the coronavirus disease 2019 or COVID-19 pandemic continues to disrupt healthcare systems, economies, and social behavior worldwide despite vaccine advancements. Therefore, preventing community spread of COVID-19 is still a major challenge. To reduce or prevent community spread, both medical and non-medical strategies are being utilized, and many of these approaches on this chart are being employed. COVID-19 testing plays a major role in these specific non-medical countermeasures to better inform individuals and health to mitigate disease transmission. There are two major categories of COVID-19 diagnostic tests for active infections. The first one is molecular viral RNA, and the second one is rapid antigen test genes. Molecular tests are extremely sensitive to amplification of viral genetic material, but they are resource and time intensive to process. There are also limitations due to cold storage requirements and supply chain issues as well. On the other hand, rapid antigen tests are simple to administer, can provide point of care results within 30 days, less expensive to produce than molecular tests. While existing rapid antigen tests aren't as sensitive as molecular tests, they can be useful as high throughput screens that can mitigate transmission. To potentially approve rapid antigen tests, I'm investing the potential of using DNA aptamers, antibodies in these rapid diagnostic tests for SARS. DNA aptamers illustrated DNA that forms secondary structures that allow specific recognition and binding of target molecules, including proteins. Aptamers can achieve binding to target molecules with similar affinities to antibodies, but they also have many other advantages, including cheaper costs stability. To use aptamers in rapid antigen tests, we have to select for one that targets the SARS-CoV-2 virus. We're interested in targeting the spike protein because it's already used as a recognition in existing antigen, and it's also what bodies generated against SARS binds basis for the S1 domain. The domain contains, while the S1 domain recognizes the host cell. Looking further into S1, the S1 domain contains domain or RBD, which binds the ACE2 receptor on the host, and the N-terminal domain, whose exact function is still under investigation. Now that I've introduced all the pieces, we initially for the SARS-CoV-2 aptamer diagnostic project, which was one, to find a high binding affinity aptamer that could be on par better than antibodies, find one that specifically binds to SARS-CoV-2 similar proteins, and the third is to detect virus at clinically relevant concentrations. For aptamer sequences, we use the library selection method called systematic evolution of ligands by exponential arrangement, or SELECT. Our SELECT protein targets for selection. We use the S1 protein and the S monomer highlighted here in this box as positive selection targets. For negative selection, we used the non-relevant proteins, bovine serum albumin or BSA, to remove non-specific binders, and in later rounds, proteins from related coronaviruses, MERS-CoV and SARS-CoV, in order to enrich for more specific binders of SARS-CoV-2 S1 protein. After 12 rounds, we sequenced and 2 motifs were enriched in the penultimate onto motif 1 by, and it occupied the majority of the top 100 sequences and represented more than 60% of all reads. We realized the binding of these sequences. First, I tested whether motif 1 could bind, highlighted here in both the shades of blue, which we expect because we used S1 protein in the selects. And by flow cytometry and determined the binding affinity with biolayer interferometry, or BLI. We observed binding at concentrations as low as 6.25 nanomolar and calculated a KD of about 40 nanomolar. Next, we location of motif 1's binding site. So we tested binding to both RBD and NTD, which were the 2 halves. We found that motif 1 bound the NTD, which is highlighted here in blue on the left cartoon, and they gave about 60 nanomolar, which is similar to some antibodies. And we did not observe binding of motif 1 to RBD. Therefore, after we uncovered the binding location of motif 1, we renamed the instance for SARS-CoV-2 spike protein and terminal domain binding aptamer 1. And therefore, on our goals, we were able to meet the first one finally, which was discovering a high affinity aptamer. And so further, we wanted to characterize SNAP1, we tested binding of SNAP1 to 3 different proteins, SARS-CoV-2 S1 protein, which was used at this positive, and SARS-CoV and MERS-CoV S1 protein, which were used as negative selection targets. And that SNAP1 only bound to S1 protein, and that non-specific aptamer didn't bind any of the proteins either. And so this supports our second goal, which was specific binding of SNAP1 to SARS-CoV-2. To further characterize how SNAP1 binds as with Ha in the Wiesler lab, also at UW, to take cryo-electron microscopy images and construct a 3D model. In this image, we see that SNAP1 in purple, exclusive entity of the S protein highlighted in the lighter blue, at a recently reported antigenic supersite on the NTD. This epitope is binding SARS-CoV-2. Now that we thoroughly characterized SNAP1 in 2 proof of concept virus detection systems. The first one here that I'll show you is an aptamer modified SNAP1 instead of antibody for capturing the enolate. We're able to specifically detect as low as 10 picomolar of 100,000 UV inactivated SARS-CoV-2. Both non-specific aptamer and high concentrations of produce a signal, which further demonstrates the specificity of the assay. Thus, for our goals, we met the third and final goal of being able to detect virus, as well as continuing to support high affinity and specific. Lastly, we use SNAP1 assay, which aligns more closely with our vision of employing SNAP1 in rapid diagnostics. In this assay, we use a lateral universal kit to demonstrate rapid detection. We added 2 differently labeled aptamers, one either labeled with FITC or biotin, to a sample solution containing the analyte, and then we dipped the test strip in. It has gold nanoparticles in it, and with the solution due to capillary action. The gold particles that bind the complex accumulate at the test band, while unbound gold particles accumulate at the control band. And using this to detect S protein at as low as 4 nanomolar in concentration, and also UV inactivated SARS-CoV-2 at about 10 to the 7th copies. So therefore, to summarize our talk today, my talk, the first checkpoint that we managed to complete was high affinity, and we had it around 60 nanomolar KD. We were able to achieve specific, it only bound SARS-CoV-2 as one, and SARS-CoV-2 spike proteins in general, and not MERS, and it didn't bind lentivirus. Also able to detect virus at 10 to the 5th copies with ELISA and 10 to the 7th copies per mil with lateral flow. And clinically, clinical samples are around 10 to the 4th to 10 to the 10th copies per mil. So so our assays are potentially useful for those kinds of samples. And today we're continuing our work in several directions. We're selecting for variant S proteins such as the alpha variant and the delta variant. And we're also applying SNAP1 and other aptamers that we've discovered to other diagnostic systems such as aptamer virus and antibody-free lateral flow assays. So thank you for our talk, and I'd like to thank Professor Susie Pun for supporting this work, and our lab manager and research scientist for supporting all aptamer work in our lab. There's Ha and Dr. Wiesler. All right, thank you.
Thank you, Lucy, for we had one in the chat that's asking properties of the aptamers when I guess you discover them. So the binding affinity, etc., can be translated to the in vivo easily. And I guess I could rephrase this like whether it translates from in vitro to biological like detection, right? Or to mixing it with biological sample.
Yeah, that's a great question. We first attempted to treat cells with we first tried to use this in viral inhibition using an in vitro model, and we didn't see any inhibition. We also tried other aptamers also reported in literature. We did not find too much success with that, so we moved on to detection. But that is something we were initially interested in.
Thank you. So we are a bit late on schedule. Go directly to the next talk from James that's going to discuss and present us his progress on the synthesis of DNA and RNA using mechanochemistry.
Hi, everyone. Thanks for the intro and thanks for the opportunity to be here to present my work on scalable synthesis of DNA and RNA oligomers using mechanochemistry under the supervision of Dr. Dama at McGill. So as we know, with the growing field of oligonucleotide therapeutics, there's also a huge demand now for synthetic oligonucleotides. Fortunately, with current methods for oligonucleotide synthesis on polymer supports using phosphoramidite chemistry, we have extremely high coupling yields, up to 99%. The manufacturing process is automated, and we can manufacture oligonucleotides on a large scale, up to kilograms. Unfortunately, as Dr. Dama alluded to earlier today, the solvent footprint for the oligonucleotide synthesis is huge, with thousands of liters of organic waste produced per kilogram of a typical 21-mer oligonucleotide synthesized. So the aim of my project was to try to address this major issue of oligonucleotide synthesis to make it more economical and more environmentally friendly. This brings us to the field of mechanochemistry. Mechanochemistry is a relatively new discipline in the field of chemistry where energy to provide the energy for reactions as well as provide efficient mixing for chemical reactions. The advantage of mechanochemistry is that, as you can see here, we often perform the reactions completely in the absence of solvent or in very small amounts, stoichiometric amounts of solvent to facilitate with mixing. Shown here is a typical vibration ball mill or VBM apparatus, which you can set the frequency. And as you can see, the reaction can take place here between solids without the use of any. So having this relatively new tool in chemistry at our disposal, we decided to see if we could apply mechanochemistry to the synthesis of oligonucleotides. When we first set out, we actually approached the problem via the H-phosphonate chemistry. This is due to the increased stability of H-phosphonates as compared to phosphoramidites, and we thought that they might be more amenable to mechanochemistry. So starting here with the 3' H-phosphonate nucleotide, we were able to couple it to a 5' OH nucleoside in the presence of 5 equivalents of adamantane carbonyl chloride, which is our activator, and a very small amount of pyridine, just 10 equivalents to act as a base and to help with mixing. And as we mixed it in the vibration ball mill for 15 minutes at 25 Hertz, we were able to form these H-phosphonate diesters. Fortunately, we're able to really easily monitor these reactions by phosphorus NMR, where we have a distinct chemical shift for the H-phosphonate monoester 1C here coming about 3 ppm compared to the H-phosphonate diester, which comes as a pair of diesteriomers around 8 to 10 ppm. One of the issues we found, though, after combing through the literature and observing it ourselves, is that H-phosphonate diesters are actually relatively unstable, and particularly they degrade, as you can see here in the third spectrum, under treatment of basic aqueous conditions. And we were really just having a hard time isolating these compounds, so we decided to go with a different approach, where we basically oxidize the H-phosphonate linkage to this thiophosphoric ester in a strategy most recently used by Dr. Colin Rees, where we use N-phenylthiophthalamide, or PTP, as a sulfur transfer reagent in one equivalent with 5 more equivalents of pyridine milling for 15 minutes at 25 Hertz to convert the H-phosphonate diester to this thiophosphoric ester. Once again, we monitor this by phosphorus NMR, where we see a new set of peaks at around 24 to 25 ppm, and we were able to isolate these compounds by column chromatography. Additionally, we were able then to do these 2 steps, the coupling of the nucleoside with the H-phosphonate, followed directly by in situ oxidation and protection of the phosphorus linkage as a thiophosphoric ester, and we were able to isolate these in pretty good yield, 76% over 2 steps. Finally, from here, we were able to deprotect the dimer fully by removing the 5' DMT group and then converting the thiophosphoric ester to the phosphodiester backbone, and then finally treating with ammonium hydroxide to remove the 3' levulinyl group and any base protecting groups. And we could see also these 2 peaks around 24 ppm from the thiophosphoric ester then turn into a single peak for the phosphodiester around -1 ppm. Having now gotten nice optimized conditions for the coupling and protection of the H-phosphonates using mechanochemistry, we could set out to establish some nice synthetic strategies to try and synthesize longer oligonucleotides. So starting here from the same starting materials, repeating the experiment with all 4 bases, we were able to synthesize these dimers in generally good yields in 2 steps using mechanochemistry. So from here, we have a few different options. We can then remove the 5' DMT group using TFA and DCM and methanol to get these dimers with free 5' OH groups, and we're able to isolate these in good yields. Alternatively, we can remove the 3' levulinyl group to get these dimers with free 3' OH groups, and from here, we can then treat them with diphenyl hydrogen phosphite to synthesize a dimer with a 3' H-phosphonate. Alternatively, one of the strategies that we went for is we synthesized now a phosphoramidite at the 3' N, and then we have these stable dimers, which can have a really nice application in solid phase synthesis, especially for the synthesis of longer DNA sequences. So having all these different dimer blocks in hand, we set out with a few different strategies. So we could approach it just with a monomer coupling approach, where we just grow an oligonucleotide chain, 1 nucleotide at a time, So basically taking that dimer with the free 5' OH group and coupling it with another equivalent of the H-phosphonate monomer under the exact same conditions. And we were able to isolate these trimers in good yield. Alternatively, with these dimers and these dimer H-phosphonates, we're able to grow these in a dimer block coupling approach where we grow the oligonucleotide chain 2 nucleotides at a time. So here we have the 5' free dimer coupled with the dimer with the 3' H-phosphonate under the same conditions to synthesize a tetramer in good yield as well. Finally, we then remove the 5' DMT group of the tetramer and then couple it with another equivalent of the dimer H-phosphonate under the same conditions, and we're able to synthesize a hexamer also in good yield. And one of the really encouraging things about these results is that as you can see, the yield of the coupling doesn't really drop off. So we have a pretty standard yield that we get for the coupling, even as we go to longer and longer strands. But we didn't want to stop here with just DNA, so we decided to see what other chemistry we can do here. So we then moved to RNA chemistry instead of just DNA. So now we have a 3' phosphonate RNA with a TBDMS group protecting at the 2' end, and now our nucleoside with the free 5' OH group has the same 3' levulinol protecting group, but instead of having the TBDMS group, we have the TIPS groups, or the triisopropylsilyl group, and the use of that at this end is going to come clear when we synthesize the dimers. Of course, the next step is to do the same type of block coupling that we did with the DNA chemistry, but now with RNA. And the reason we have to use the TIPS group is because if we had a TBDMS group at this 2' position, upon removal of the 3' levulinol group, we would likely see isomerization between the 2' and 3' positions, whereas the TIPS group has been shown not to isomerize. So that work is in progress right now, and we're hoping to get some longer RNA sequences shortly. Additionally, we can synthesize not only phosphodiester backbones, but by a simple switch of the sulfur transfer reagent here, instead of having the thiophenyl group, we switch it to a cyanoethyl group on the sulfur here. And of course, this is reminiscent of the cyanoethyl protecting group used in traditional DNA synthesis. And from here, we can see that if we were to remove the cyanoethyl protecting group via beta elimination from treatment of base, we could get to the phosphorothioate backbone. Of course, this is a hugely important chemical modification in oligonucleotides. And additionally, we can also do the same type of reactions with 2' O-methyl nucleosides, such as this one here, where we have the same type of reactions taking place, except we have the 2' O-methyl H-phosphonate, and we're able to isolate these in good yield as well. So in conclusion, we the first mechanochemical synthesis of DNA oligomers, and we were able to cut our solvent use by up to 90% during the reactions. We have a few nice strategies, phosphonate chemistry, where we can synthesize oligomers via monomer or block coupling approaches. We were able to synthesize dimer amidites, which are highly valuable for solid phase synthesis. And of course, we were able to show RNA, 2' O-methyl, and phosphorothioate backbones possible. That we have currently in the pipeline, we'd like to continue protection conditions to show we can fully deprotect these oligomers efficiently and isolate them. And of course, we'd like to synthesize longer oligomers, longer than a hexamer, up to more therapeutically relevant lengths, and especially chemistries not have just solely DNA oligomers or RNA, but combine them in different combinations. And finally, as there was some question, we're looking at improving the purification techniques. As I said, we purify these by column chromatography. Of course, that uses a lot of, definitely looking at some other methods where we can purify these by precipitation or some other methods. Thank the Dama Lab and Dr. Dama for his supervision, as well as for their help with the mechanochemistry experiments, as well as NSERC and McGill Sustainability Systems Initiative for funding. And I would be happy to answer questions that you guys may have. Thanks.
Okay, thank you so much, James. It's always great to hear. and how far you're progressing with the length of the oligomers. We have a question in the chat from Sir, and he asks, have you tried to oxidize the internucleoside H-phosphonate with just iodine water? So I guess referring to the traditional oligonucleotide cycle rather than with derivatives you mentioned.
Yeah, that's a good question. We haven't done too much oxidations. I think we would probably run into issues with the other steps if we just had the native phosphodiester backbone present throughout the entire sequence during all of our coupling. The thiophenyl group, pectin group as well.
Okay. And you guys are welcome to continue this discussion in the chat. Thank you, James. So we will continue on to our next, and it is Prabhuda Mukherjee, going to talk about differential uptake of antisense oligonucleotides in mouse hepatocytes and macrophages revealed by simultaneous 2-photon excited fluorescence and imaging. Okay.
Thank you for the opportunity for this talk. So my name is Prabhuda Mukherjee, and I am an optical imaging scientist here at the Center for Optical Molecular Imaging. It's a GlaxoSmithKline Center. So we are located at the University of Illinois at Urbana-Champaign. And today I would like to present our multimodal imaging studies that includes 2-photon excited fluorescence and coherent Raman that we employ to study or investigate the differential uptake of PSASOs in mouse hepatocytes and macrophages. So next slide. So as we enter the next paradigm of drug technology in the form of PSASOs, so we are very attracted to it because it can actually identify viral RNAs or DNAs, and actually they can cleave them or basically help any disease. So our interest is in hepatitis B virus. And so one of the challenges actually was how to specifically target hepatitis. But like, and it has demonstrated pretty 3-4 years before that, if you a ligand called the GalNAc to this PSASO backbone, that for systems like if you dose them, there is an 8 to 10-fold increase in the efficiency of the GalNAc PSOs with respect to the regular PSOs, regardless of the dosing. So this was very attractive. And following this strategy itself, they were like to based on the GalNAc ASOs and regular ASOs, when they were like healthy human, the GalNAc expected because the regular healthy human subjects, the GalNAc delivered more than the regular. However, when they were like patients with hepatitis B virus, the regular PA performed the GalNAc. So this was the apparent contradiction brought back us again to the drawing room to actually design the experiment. Why doing this? To a wise approach, at which we started with the cell culture studies, regular hepatocytes and mouse macrophages to see if we can actually reproduce the first result on the mouse studies that has been Prakash et al. For the ASOs against the SR. So the main target is using a tagged PSASO fluorescence marker attached, like the Alexa 488, wanted to transfect them to different cell types, like liver cell types, like macrophages or hepatocytes. These GalNAc is actually, has a strong affinity for these SGR pathway. Actually binds to the cell surface, and some kind of an expecting that when how do we of these the galenic tag PSOs in regular hepatocytes, and not only see a comparative uptake between the regular hepatocytes. Apart from that, if there is a macrophage, and we actually compare the uptake rate between the hepatocytes and macrophages when. So in this slide, I would like to acknowledge Ionis Pharmaceuticals, generously giving us all the PSASOs with GalNAc and the Alexa 480 tag for our and also were performed here at the GSK Center for Optical the University of Illinois. So into the study, let me just describe the system that we use for our study. Multimodal imaging microscope that we had developed here in our lab, and it has the top right, bottom right corner microscopes that are being fed continuously by the laser system. This is the microscope, which is a multimodal imaging microscope that we are using, and you can see the green arrow, the channel that we'll be using for our Alexa 488, and this is an inverted microscope. And the other one, so we have a coherent anti-Stokes Raman microscope. Getting into more details, so just briefly jot down a little some points about this microscope. Here we have two beams shown in are overlapping on top of each other, and they are being sent to the sample. And the difference of the frequency of these two beams, they create vibrational, they actually monitor the vibrational frequencies of molecules. They can be detected mode as seen from PMT1. But we can also measure the two photon fluorescence simultaneously in the epidirection. So these are the strategies that we are going to use to measure our measure the cells, these ASOs. An example, stars image excited emission at the same time, co-registration. And so these were rat hepatocytes. Actually, there is a GalNAc ASO, Alexa 488 time like 3 hours from the transfection. So you can see is the cars where we are mostly highlighting the lipid droplets as shown in white. Channel is the Alexa 488 tag, and it are cells that have these really red clusters, and we that dead viability. And when we opposite image, what actually is interesting, a lot of clusters both in the green and the red channel mutually exclusive, which actually leads to the conclusion that these in the hepatocytes are actually not participating in studies of the GalNAc tag is. So before set of experiments, we wanted to do a concentration dependent study of these GalNAc ASO macrophages and hepatocytes. So this is the how different the images are. The concentration of the ASOs. So this is 500 nanomolar and 5 micromolar. So we always want a quantitative estimate, and these are the we actually estimated the amount of that are being uptaken by the macrophages. The standard deviation that we obtain from individual cells. So in the next slide, this is the for GalNAc ASOs. Now for the 500 nanomolar and 5 micromolar case, you can see there is no very suggestive difference, and that is exactly what we found in the quantitative estimate, that there might be a saturation concentration after which these ASOs are not uptaken anymore. And so this is I'm showing you the uptake mechanism or uptake video. It's a time lapse of these hepatocytes that are with the GalNAc ASOs and separately. And so we are measuring from 30 minutes 1 hour 30 minutes, and the frame rate is about 30 seconds. So what stands out, see the apparent change of PSOs in GalNAc and the non-GalNAc one. So whereas in the see a peripheral kind of uptake, so the membrane kind of lights up, and then the thing starts to dissipate inside the cytoplasm. In the case of PS, here's the region that is getting means that part of the cell is polarized uptake of these ASOs. So this is what we are actually kind of want to say, like it's a peripheral versus polar uptake of GalNAc ASOs in mouse hepatocytes. In contrast, when we look at the macrophages itself, they are the same, as expected, because macrophages particles in any kind of endocytosis mechanism. And to not to our surprise, macrophages for both when transfected with both the GalNAc ASOs, they are showing a very similar, here. So to quantify for panel, I'm showing the rate of uptake for the mouse macrophages for the PSASO and the GalNAc ASO, and I'm showing you the uptake pattern for the PSASO ASO for the hepatocytes. As you can see, as expected for the macrophages, we don't have any deviation. It's like slight changes for the PSASO. Actually uptakes a little bit more, but for the GalNAc ASO, we see almost a factor of 3 to 5 change in the uptake pattern, and hence the amount of ASO. So this is very, very similar to what we anticipated from mouse tissue studies that has been done previously. And so we were happy just to reproduce this result just from the cell culture. In conclusion, what these multimodal imaging studies gave us an insight as to the uptake rate of the uptake rate of the PSASOs in mouse macrophages. For the GalNAc uptake compared to the polar distribution of untargeted PSA. And so we are hopeful that going forward, imaging techniques can help us unravel other studies, such as go to future. We can study the complex in liver model that has cellular co-cultures that have been transfected with these ASOs to actually identify different cells. And then maybe we can, in the next meeting, I can present tissue studies where we can use the multimodal to identify oligo in tissues. So with that, I will acknowledge my team, optical molecular imaging, like Steve Hood from GSK, who helped me design the studies. And lastly, my advisor, Gopart, head of the Biophotonics Imaging Laboratory, for all his support. Thanks for your audience, and I can answer any questions if you have. I think you're muted.
Orly.
I'm sorry. I realized I was muted. Classic problem, I guess. I'm really sorry to everyone. So thank you for your really, really nice talk. It was really... we got one question for you from Jan Wheeler, who is asking about whether this is observed only for antisense oligonucleotide, or did you also see it happen with GalNAc conjugated siRNAs?
We observed it for both. For both the GalNAc and the antisense. Here, it's a representative picture for the GalNAc, but I think we have observed it for both the GalNAc and the regular PSO.
Cool, thank you. I guess there will be probably other questions for you on the chat, so feel free to ask more questions to Prabuddha, and Prabuddha feels free to answer. We'll go to our next speaker, who is Ala from Duke University. And she will tell us about investigating the impact of epitranscriptomic modification on RNA samples.
Hello, everyone. Thank you so much for the opportunity to present. I'm happy to share the work that I have been doing at Duke University during my postdoc to understand the impact of epitranscriptomic modifications on RNA ensembles. During the past 2 decades, studies have emphasized the dynamic nature of RNA, showing that they do not adopt a single static conformation, but rather an ensemble of many interconverting conformations. And the conformation landscape of even a simple RNA, such as HIV-1 TAR shown here, can actually be highly complex, with a single secondary structure dominating the ensemble of free TAR in solution with a population of 80%. Additionally, there are low populated non-native secondary structures with populations less than 10%, and these forms on the micro to the millisecond time scale and differ with respect to base pairing around the bulge and apical loop. And because these conformations have markedly different structures than the ground state, they will have significant difference in their biological activity. And the population weighted average of all of these conformations is what we refer to as an RNA ensemble. And cellular conditions and other perturbations can affect the relative populations of these conformations rather than create new ones. It reminds me of a quote I saw in one of Professor Steit's papers. She was referring to RNA-RNA interactions, but fits here, which is ignoring this RNA landscape is like walking blindfolded into a beautiful wilderness. And the reason I think this is true is because the functionally active conformation of a certain RNA may not always be the most populated in solution, and you might miss it. And now, that many RNAs are post-transcriptionally modified, we are interested in investigating how do these modifications alter the ensemble of an RNA and therefore affect its biological activity. To date, there are more than 150 epitranscriptomic modifications that have been discovered. The majority of these modifications we are interested in a 2'-O-methyl ribose modification, which we refer to as NM. It is one of the most common modifications in the RNA world and, of course, in the oligonucleotide therapeutics world. NM occurs at the 2' hydroxyl moiety of all 4 nucleosides, A, U, G, and C, in addition to nucleosides with modified nucleobases such as M6AM shown here. Giant advancements have been made in the field of epitranscriptomics, where readers, writers, and erasers for some modifications have been discovered, such as for M6A, for example. However, there are still several modifications, unknown reader proteins, unknown biological function or mechanism of action. So how do these in general exert biological activity? The first mechanism that we can think of is recognition by reader proteins, which will then splicing, export, etc. The second mechanism is steric clashes or disruption of interaction. The third mechanism, which we are interested in, is redistribution of the RNA ensemble, where a modification is a conformational switch to stabilize or destabilize certain conformations. For example, here, the addition of M6A reduces the energetic cost of conformational change, opening up the RNA duplex, allowing the protein to bind to the single-stranded region, which was otherwise not accessible in the absence of M6A. And modification found in coding and non-coding RNAs, such as ribosomal RNA, snRNA, and more recently discovered internally in mRNA regions, and has been linked to several biological processes, and such as the ones listed here. NM is one of the RNA modifications that does not crick base pairing phase, and therefore making its contribution to structure and function harder to enter. Introducing NM to single-stranded RNA leads to stable RNA duplexes, and on average, each NM modification leads to 0.2 kcal per mole stability. Additionally, the ribose moiety is intrinsic and is in equilibrium between C2' endo and C3' endo sugar puckers. The introduction of 2'-O-methyl shifts the equilibrium more towards the C3' endo conformation, which is the canonical conformation in A-type complexes. So given that NM stabilizes RNA duplexes and favors C3' endo sugar pucker, we hypothesize that NM could potentially use these properties to modulate some. And to test this hypothesis, we decided to use HIV-1 TAR as a model system, since it is one of the very few RNAs for which a detailed and comprehensive energy landscape is available. The ground state of TAR consists of species separated by a trinucleotide bulge and a hexanucleotide apical loop. Apical loop residues are flexible and possess a C2' endo sugar pucker, while the helical regions possess C3' endo sugar pucker. Studies from El-Hashim relaxation dispersion experiments have shown that TAR exists in dynamic equilibrium non-native conformations that we refer to as excited states. The first excited state forms through the localized zip loop and the formation of two non-canonical mismatches. This excited state is considered to have a high population, however, has a very short lifetime, and that's why it is hard to detect using techniques. On the other hand, excited state, ES2, forms through a tree all the way through the This excited state has a longer lifetime, however, it has a very low population. And for the purpose of this talk, I will focus on how modifications affect a ground state to ES1 transition. So here I forgot to mention that the transition ground state to the excited states have been accompanied by changes in sugar pucker, where you can see here the blue flexible unpaired nucleotides will end up being C3' endo in the helical regions of an excited. Given that the excited state, ground state to excited state transitions are accompanied by these sugar repuckering events, we reason that NM, when positioned in these nucleotides, could potentially redistribute the ensemble and stabilize the excited state. And therefore, we introduced NM at A35 here to stabilize ES1. And to gain insights into how NM affected the ground state to ES1 transition, we performed NMR relaxation experiments on isotopically site-labeled modified and unmodified TAR samples. Unfortunately, due to the limited time, I will not be able to go into the details of the relaxation dispersion technique, but I'd be happy to go over it in the Q&A session if anyone is interested. But in general, when we run the relaxation dispersion or RD experiments, we get dispersion profiles that look like this. And then from fitting this data, we can actually get the population of the excited state, the forward and backward rate constant, and we can determine the chemical shifts, which will help in determining the structure of this excited state. As you can see here, that after introducing NM to TAR, we can see that the profile, the RD profile is increased. And the fit revealed that NM increases the population of ES1 by twofold. Interestingly, NM was found to have a minimal effect on the forward rate constant, which is the ground state to excited state. However, it reduced the backward rate constant, that is the excited state to ground state, by almost twofold, and therefore enhanced the population of these newly formed non-canonical base pairs and the excited state. And great, now we know how does NM affect the ground state to excited state transition. What about other? So as I mentioned, NM stabilizes duplexes by approximately 0.2 kcal per mole per modification. On the contrary, for example, M6A destabilizes duplexes by approximately 0.5 to 1.7 kcal per mole per modification. And the reason for that is that in order to be able to form Watson Crick, methyl group here has to adopt the anti-conformation, and this leads to steric hindrance. So in a way, M6A is acting in an opposite manner to NM when placed in RNA duplexes. So we wondered, how will M6A behave in our TAR model system? My lab mate Bailey introduced M6A instead of NM at this position here. And as you can see that the RD signal is almost totally destroyed when M6A is introduced. And then when we fit this data, we determined that the population of the excited state is now reduced to 1%. And interestingly, M6A affected the exchange 150-fold, mainly by slowing forward rate constant, which is the exact opposite of what we observed in the NM case. So that was pretty interesting. And then we were curious to determine 6AM due to the ground state to excited state transition. Will M6A and NM in M6AM have an additive effect. And indeed, when I performed these experiments, you can see we got a very nice RD profile. So it kind of revived the effect of M6A. And fitting this data showed that we indeed have an enhancement by approximately 5%, and the results are in good agreement with an additive effect for NM and M6A. And I find this is really fascinating because these change in rate constants from one modification to the other can probably hint us to why does nature choose one modification at a certain position in an RNA, probably to tune RNA-RNA interactions during the several biological processes that need RNA rearrangements. And with that, you can see that small local changes to the ribose or nucleobase can induce large global changes in structure and alter the RNA landscape. For example, I showed you here today one example where a modification stabilizes the excited state while the other one stabilizes the ground state. And but there's still a lot of modifications to discover. And with that, I would like to thank my supervisors and the organizers for the opportunity to present, and I'd be happy to answer. Thank you very much.
Hey, thank you so much, Hala. It's always great to hear from you. We have a question in the chat from Dan O'Reilly, and he says, fantastic talk. Really, the 2' methyl is changing the lifetime and abundance of structures, as a structure plays a big role in whether we can effectively silence the RNA. Anyone looked at the relationship between this modification and silencing capability using ASOs?
So of course, this modification has been introduced in most of the therapeutic strategies. So that's why it's interesting, because it's very interesting to take into account how this affect the kinetic rates. For example, when you want to do gene silencing, the modified RNAs should, it might affect basically the hybridization kinetics, and that's something that we're working on right now. So yes, so this modification affects the dynamics, and the kinetics for therapeutics is actually very important, and that's what we're working on now.
Okay, Dan, I hope that answers your question. And then also, Hal, I have a question, something else unrelated, but how do you envision will knowing the excited states or energy landscape of the RNA help us design drugs to target a certain RNA?
That's a great question. Thank you, Leonora. So basically, it's very important RNA as an ensemble and not a static structure, as I mentioned in my very first slide. The active conformation, for example, in the TAR case is usually the base triple conformation, which is less than 0.1% populated. And what happens in real systems is that when the super elongation complex binds, for example, to TAR, it shifts the equilibrium more towards this very rare conformation in solution. So if we didn't know this landscape, that's the active conformation, and then we will not be able to actually target it. And on another note, it's really important to know all of these conformations when you are trying to design target an RNA. And studies from Hashim's lab have shown that if you view RNA as an ensemble and try to dock different molecules to the ensemble rather than just a single, get better score, better luck in designing molecules that actually target these RNAs.
Okay, that's amazing.
You're welcome.
We will move on to our final talk of the session, and that is coming from Albert Kamanzi, and he will simultaneous single particle measurements of size and how they give new insights into the structure of drug delivery nanoparticles.
Hi, everyone. Thanks for my talk. My name is Albert Kamanzi. I'm a postdoc at UBC, and I'm happy to present a single particle image that we are using to study lipid nanoparticles. And with its mathematical parameters, we are able to get structural information from our measurements. So why are we interested in lipid nanoparticles? As you know, generic medication have shown great promise to treat patients as well as design new. However, delivery of these drugs is still a challenge. Lipid nanoparticles have shown great promise, as shown in the current pandemic. Hence, understanding their properties and their formation can design better drugs. Basically, we are interested in looking at lipid nanoparticles, understanding their structure as a function of different conditions, specifically particles that are used for the current vaccines are known to change the structure as you change buffer conditions from lower pH to higher pH, as well as they form different structures when they are loaded versus when they are not loaded with drugs. We're using to study particles, and CLIC is basically an add-on to a microscope, which allows us to track single particles in microwells and do particle tracking on single particles. And from these single particle tracking, we can build MSD curves of the diffusion of these particles. And from the MSD curves, we can extract size and loading information of these nanoparticles. So CLIC is made up of a deflectable flow cell to trap array of nanoparticles in wells. And from this parallel array, we can build large statistics from our measurements. So we start by, we first use our technique to validate our measurements by looking at non-beads. We specifically looked at polystyrene beads. By sizing the polystyrene beads, we basically characterized our system, and by doing single particle tracking of the beads and building distributions of MSD as well as size distributions from the beads, we validated our method by doing measurements at different, basically different confinement heights of our wells. And we know that when you do diffusion measurements near surfaces, you get surface interactions which affect the diffusivity you measure. Hence, by doing measurements at different heights, we are able to correct for the confinement effects. And with these corrections, we're able to reproduce the known size of the beads. So once we validated our method, then we went on to study lipid nanoparticles. Again, doing single particle tracking of these lipid nanoparticles. Specifically on these slides, we are using unloaded lipid nanoparticles, looking at both lipid nanoparticles at pH 4 and at pH 7. And then from these size distributions, as well as in parallel, like basically each measurement, we're able to get a size and intensity information of the single particles. And by correlating the two distributions, we actually are able to produce these plots, which allow us to correlate the size and intensity, hence loading of these nanoparticles. And from these correlation plots, we can actually get structural information. For example, on the left side, you have lipid H4, are labeled with lipophilic dyes, which are known to insert themselves into the surface of the nanoparticle. And hence you expect a shell-like distribution. From our measurements, knowing that we expect a shell-like distribution, we expect the scaling of the correlation between intensity and size to follow a scaling of power of 2. And that's what actually we get in our hence we get structural information, the expected structural information at pH 7. However, at pH 4, the scaling we got was much higher than 2. It's around 2.3. And this can be explained by the fact that at pH 4, the nanoparticles actually form these bilayer structures that are filled with an aqueous volume in the center. Hence, this allows the DII dye we used to actually also reside on the insides of the bilayer. And hence, we get a scaling much higher because you're starting to fill the volume of the nanoparticle with the dye. And this is in agreement with the cryo-EM measurements done on the same sample. Another type of measurement we do is combining click tracking with this time we are looking at lipids that are loaded with drugs, and the drugs are tagged with a fluorescent dye, and the labeling ratio you expect to have like a single labels per particle. You can actually perform photobleaching measurements, and from the photobleaching measurement count the number of bleaching events, and from these bleaching events you can actually count the number of in a particle. With our measurements, we looked at 3 different labeling fractions, and with our measurements, we're able to basically labeling ratio we expect from these measurements, again showing that we can do single loading measurement molecule resolution. Looking at the loaded nanoparticles, this time we label the dye, we label the drug instead of the nanoparticle itself. And by looking at the size and intensity scaling again, we can look at the distribution of the drug inside of the particle. With these measurements, we're expecting to see a scaling of 2, assuming the drug basically follows a shell-like structure inside of the particle, or a scaling of 3, assuming that the drug basically resides in the entire volume of the particle. However, our measurements were not correlating with either case, and this could be explained by the drug inside of the particle basically forming, not following the scaling of volume or the surface of the particle. Basically, we expect that the loading of the drug is more complicated than the simple, the 2 simple cases we are expecting. Hence, again, with our measurements, we can probe the distribution of the drug inside of these particles. So with this technique, we are able to perform single particle imaging. We're able to make single particle measurements on lipid nanoparticles and build distributions with single molecule resolution. And we use this technique to look at to study lipid nanoparticles, get their size distributions and loading distributions. And from the correlations of these measurements, we can get structural information of these lipid nanoparticles. We are interested in looking at the dynamics. On this slide, I'm showing you a movie of lipid nanoparticles that are actually, again, trapped in the microwells. And by changing the buffer, you can actually directly watch as the lipid nanoparticles are fusing with each other. So we are interested in studying the dynamics of these lipid nanoparticles as a function of the changing conditions. And we're interested in of large biomolecules like messenger RNAs or self-amplifying RNAs in these lipid nanoparticles. And we would like to go even further and look at how these drugs, lipid nanoparticles, deliver drugs in cells by doing live cell imaging. I'd like to end by supervising Sabrina Leslie, everyone in the Leslie Lab, and all our collaborators. I'll be happy to take any questions.
Okay, thanks, Albert, so much for your talk today. I have one quick question for you. Did you look at one type of sRNA, or have you looked at like different sRNA with different modification and how modification on the SI would affect the information you're loading on those lipid nanoparticles, etc.?
That's a good question. So the nanoparticles we used are actually based on the formulation for the Onopatro drug. We looked at just one drug type, but yes, we are interested in looking at like varying the drugs actually affect the nanoparticles.
Cool. I can ask one more question, I think, from Annabelle, who is asking, do the nanoparticles behave the same when isolated versus in mixture with other nanoparticles?
Mixing different nanoparticles or particle versus particles in a.
I guess probably one versus mixing them all together.
Yes, so they don't. Okay, so we have different nanoparticles. We are just looking at like the drug for now. But if multiple particles in a well, the buffer, they do fuse, like I showed in the movie. But other than that, they don't really seem to interact. Like basically, they are freely diffusing with within the same well.
Thanks a lot. So I guess there was one more question, but you can go on the chat and please feel free to answer in chat a little bit more. And so we're gonna, I guess, end the session of today. Like before ending the session, to thanks all the speakers, nice research, and everyone who participated to the conversation. It was really nice to hear everyone's thoughts. So we'll share the link to vote for your favorite talk among the young speakers on the chat, and I think you'll also receive it by email. And it's now time for the part of this. We'll give everyone 5-minute break to grab a snack or drink, and then feel free to join us on the spatial. We'll post the link also on the chat, and we'll see you there.
Thank you again, everyone.